So that is now. Let's test your knowledge. It's time to test your knowledge in pyruvate metabolism. So the first question is, a well-fed individual engages in vigorous exercise that requires high amount of energy. In their muscle cells, which of the following sets of conditions would lead to maximal activation of pyruvate dehydrogenase complex? Maxima. Let's recap. A well-fed individual engages in strenuous exercise. In their muscle cells, which of the following sets of conditions will lead to the maximal activation of the pyruvate dehydrogenase complex? So let's go there. A said high ATP, high NADH, and increase in PDH kinase activity. Remember, the question is, which of the following sets of conditions will lead to maximal activation of pyruvate dehydrogenase complex? And also recall that pyruvate dehydrogenase complex is an enzyme that converts pyruvate to acetyl-CoA in aerobic respiration, the presence of oxygen. And they're now asking us, what are the factors that will stimulate this pyruvate dehydrogenase complex? You have to pass this question. So A said high ATP, and whenever there is high ATP inhibits this reaction. So this one is bad, is not part of the way. They said high ADP, high NAD positive, and increase in PDH phosphatase activities. I don't know whether it's correct or not. So they said C said low level of coenzyme A and high level of acetyl-CoA. Whenever there is high level of acetyl-CoA, what will happen? It will start inhibiting pyruvate dehydrogenase complex and start stimulating what? Pyruvate carboxylase. Therefore, this is out of the way. And D said high pyruvate concentration and increase in pyruvate dehydrogenase kinase activity. And remember that in pyruvate kinase inactivates pyruvate dehydrogenase, and when something is inactivating pyruvate dehydrogenase kinase, that means it is not stimulating it, it is not activating it. Therefore, this is out of the way. And in A said high ATP inhibits pyruvate dehydrogenase. High NADH signifies high ATP, and high ATP inhibits pyruvate dehydrogenase. And increase in pyruvate dehydrogenase kinase inactivates pyruvate dehydrogenase. Therefore, my answer is B. Why? The answer is B because high ADP signifies high NAD positive, and this high NAD positive signifies high ADP, and this high ADP signifies low level of ATP. And when the ATP level in our body is low, our body will try to make ATP, and the process by which our body tries to make ATP is stimulated by an enzyme known as pyruvate dehydrogenase, and this pyruvate dehydrogenase now converts pyruvate to acetyl-CoA, which will now enter the Krebs cycle to produce the energy our body needs. So high ADP, high NAD positive, and increase in PDH phosphatase activity. So pyruvate dehydrogenase phosphatase, what is it doing? Remember that pyruvate dehydrogenase phosphatase is what? Is removing the phosphate group from the pyruvate dehydrogenase, and when it is removing this phosphate group in the pyruvate dehydrogenase complex, it starts activating. And the question is, what is activating this pyruvate dehydrogenase? So therefore, it is pyruvate dehydrogenase phosphatase. That is the answer to that question. So explanation: high ADP and NAD positive are allosteric activators indicating low energy status. During exercise, calcium ion efflux activates PDH phosphatase, which is pyruvate dehydrogenase phosphate, which dysphosphorylates and strongly activates. What is dysphosphorylation? Removing of what? Phosphate group. Phosphorylation, adding of phosphate group to a substrate molecule. Dephosphorylation, removing of phosphate group to a substrate molecule. So dysphosphorylation of pyruvate dehydrogenase complex activates pyruvate dehydrogenase complex to produce more acetyl-CoA for the tricarboxylic acid cycle or the Krebs cycle to now produce energy. Here's the second question for you. In a patient with a genetic defect causing severely reduced pyruvate dehydrogenase complex activity, you would expect to see an accumulation of lactate. The primary biochemical reason for the conversion of pyruvate to lactate in this condition is to what? Directly produce additional ATP, generates carbon dioxide to buffer the resulting acidosis. No, B is completely out of the way. B is not even close to the option. Replenish the cellular pool of NAD positive for glycolysis. Okay, D said provide an alternative fuel source for the brain, which is wrong. So let's recap the question. So the answer is between A and C. In a patient with a genetic defect causing severely reduced pyruvate dehydrogenase complex activity, severely reduced, and pyruvate dehydrogenase complex converts pyruvate to acetic OA, you would expect to see an accumulation of lactate. The primary biochemical reason for the conversion of pyruvate to lactate in this condition is to so we're going to go for C. C said replenish the cellular pool of NAD positive for glycolysis. Remember, we are converting lactate. We are converting pyruvate to lactate. Our main major aim is to produce NAD positive that will continue to proceed with glycolysis. So be careful, the question are trickish. So with the aerobic pathway block, the cell relies on anaerobic glycolysis for ATP. So the conversion of pyruvate to lactate by lactate dehydrogenase oxidize NADH to NAD positive, which is essential for the continuation of glycolysis. A patient's liver cells are found to have abundant acetic OA from fatty acid oxidation, but the tricarboxylic acid cycle is running slowly due to depletion of intermediate for biosynthetic process. Which enzyme is allosterically activated by acetic OA to solve this problem? So go back to our playlist. We said an enzyme is activated allosterically to stimulate the conversion. It's activated by acetic OA. So which enzyme? Is it lactate dehydrogenase? No, acetic OA stimulates another enzyme because we have high yield of acetic OA. So our body will not like to produce this acetic OA anymore. So what is happening? Is lactate dehydrogenase? No, lactate dehydrogenase converts pyruvate to lactate, out of the way. Alanine transaminase? No, because it's converting alanine to what? It's converting pyruvate to alanine, so it's not out of the way. Pyruvate dehydrogenase complex. Acetic OA inhibits pyruvate dehydrogenase complex. Why? It's because pyruvate dehydrogenase complex keep on converting pyruvate to acetic OA, and there is high yield of acetic OA in our body. Our body does not need this acetic OA again, so our body starts inhibiting this enzyme known as pyruvate dehydrogenase complex allosterically. So this option is out. Therefore, the answer is what? D, pyruvate carboxylase. So acetic OA starts stimulating the conversion of pyruvate to oxaloacetate. Don't forget that. So the answer is D. Pyruvate carboxylase catalyzes the primary anaplerotic, which is filling up reaction. It is strongly activated by acetic OA, providing a mechanism to replenish oxaloacetate when tricarboxylic acid cycle intermediates are low, but fuel is what? Abundant. So this is all we have for pyruvate metabolism. So see you next time. Don't forget to like, subscribe, and hit the notification bell for more videos like this on Sagacious Learners. So next time we will start moving to the what? To the tricarboxylic acid cycle.